I guess I can go second. So good morning, everyone. Thanks for making it to the session. So my name is Jessica. I'm presenting this on behalf of Sophie Valk, who unfortunately couldn't be here today. So I'm really happy to be presenting some of the recent work that's come out of her lab, focusing on myelin plasticity and specifically in incorporating social variables in this work. All right, so when studying the brain, we think that it's important to study it within the social context and environmental pressures to which it's subjected. And this is because humans are a social species that are integrated in their social environments throughout their lives. And as a result, human brain structure and function will be shaped by this social context. However, the mechanisms that connect these aspects of neurobiology with our social environment still remain poorly understood. So if we think about this a little bit more, so social factors are integrated in human brain structure and function primarily in two ways. So first, brain structure will facilitate social interactions by providing the necessary hardware that will support social cognitive functions like perspective taking and empathy. And you can see that these regions associated with these social cognitive skills largely cover these association areas of the cortex. And in turn, the social environment will shape brain structure and function through experience-dependent plasticity. And again, different lines of research have suggested that these areas that are implicated in social cognition are also those that are most susceptible to social environmental adaptation. And one pathway to understand these interactions between brain structure, brain function, and these social environmental factors is by studying the microarchitecture of the brain, and particularly myelin. So if we look at cortical gray matter, and we know that it's organized into layers, which can be visualized using different postmortem tissue staining techniques. So these techniques can highlight the cell bodies of neurons and their myelinated fibers, which form this laminar organization of the cortex. And we know that this laminar architecture can vary substantially across regions, as was covered in the previous talk. So this can include, for example, changes in the visibility of some layers and their thickness. That's marked by the different colors in the middle panel on this slide. And this variability will allow us to differentiate areas of the cortex where transitions between regions can be sometimes very abrupt or also sometimes more graded, depending on where you're looking in the cortex. And this heterogeneity of cortical microstructure can be summarized by these broad laminar classes that are represented in the schematic here. So these configurations are expressed on this continuum of differentiation, where on one side we can clearly distinguish the appearance of the different cortical layers, but this appearance becomes less and less differentiated as we progress towards more agranular limbic cortex on the right-hand side. So what does this have to do with function and social context? Well, first, looking at cortical microstructure can help us better understand these more general principles of function and also connectivity between different cortical areas. So for example, the hierarchy of sensory-driven processing of information is anchored on these microstructural classes. And this hierarchy operates in this stepwise and distributed manner that begins in highly differentiated sensory and unimodal cortices and progressively moves towards these less differentiated association cortex that are harbored in paralimbic and limbic areas. And these regional variations in laminar architecture are also an important constraint on the topography and the strength of cortical connections. And one of the most notable examples from this line of work is the structural model defined by Helen Barbas, which lays out these principles under which two cortical regions should be connected based on the similarity of their laminar architectures. So in recent years, we've really seen the growing adoption of different MRI sequences and contrasts that are thought to be sensitive to cortical microstructure and cortical myelin, as Valerie presented earlier in the session. So these images can be used to approximate intracortical microstructural properties of the entire cortex by sampling the intensities of voxels that intersect some given surface reconstruction. And previous work using these contrasts have really recovered these systematic interregional differences in cortical myelination that we can observe between more unimodal areas compared to association cortex, with unimodal areas being more highly myelinated compared to association cortex. And these images can also provide some indications on regional laminar elaboration. So previous work from my previous group at McGill, as well as from Sophie's group, has shown how we can sample these image intensities in this depthwise manner within the cortical sheet by simply reconstructing surfaces that run between the peel and the white matter boundary and allowing us to build these microstructural intensity profiles. So now that we have these different ways to measure myelin in vivo within the cortex, we can see how these markers might change over time in different individuals. So myelin enhances signaling speed and efficiency, but another one of its properties is that it's plastic in itself and it increases during development, with association areas in particular showing the strongest change during adolescence. And at the same time, myelin will attenuate plasticity, so the more myelin is present in a given connection, the more it's consolidated. So knowing this, we can ask how the brain changes in the context of environmental adversity, like sickness, divorce, abuse experienced during adolescence. And the main question of this work that was conducted by Micah Hetwa is whether the maturation of the brain at the individual level links to more adaptive or susceptible outcomes in response to different psychosocial stressors. So what's meant here by more susceptible or adaptive outcomes? So Micah used data from the NSPN consortium, and she gathered these different adversity measures that were included in the data set that included different life events, childhood trauma, etc. And then she aimed to predict these levels of individual distress that were also collected as part of the data set based on random forest prediction models. And the difference that was observed between predicted distress by the model and true distress as was collected in the participants was this marker of outcome. So this means that if people had lower actual distress scores than what was predicted by the model, it would mean that they were more adaptive or doing better than expected given the level of adversity to which they were exposed. Conversely, if their actual distress was higher than predicted, it means that they were more susceptible or doing worse than expected. So in this accelerated longitudinal design, when looking at changes between the two time points that were collected, a higher rate of myelin-sensitive imaging changes were observed in adolescents who developed towards more resilient outcomes, and this was particularly seen in prefrontal areas bilaterally. So these microstructural changes can be interpreted as an accelerated increase in myelin in these more resilient adolescents. So next, Micah explored whether changes in structure were paralleled by changes in function. And interestingly, functional connectivity was maintained in individuals that were more adaptive, and in particular in regions that were associated with the default mode network and in frontoparietal control networks. And finally, as part of this study, Micah looked at more system-level maturation effects, meaning how structural similarity across different cortical regions changes between baseline and follow-up. So specifically here, two different modes of change were observed or were explored. So changes could be either more conservative, meaning that regions that were similar in their microstructure at baseline still became more similar over time, or changes could be more disruptive, so regions that were similar at baseline became more different or less similar over time. So here, Micah found that superior and inferior areas that are shown in red here on the left-hand side shown more conservative patterns of change. And conversely, association areas that are shown more in these blue areas rather showed more patterns of more disruptive change. And importantly, there was also this stronger reorganization with increasingly resilient outcomes. So this is the figure with the blue patches on the right. And this was particularly seen to cover these more eulaminate areas and corresponding to the default mode functional network. So to summarize this study, more resilient outcomes to psychosocial stress in adolescence was associated with greater microstructural refinement, notably involving an increased rate of myelination in prefrontal regions, as well as an increased reorganization of microstructural similarity, targeting more broader transmodal regions. However, functional networks that were associated with these subregional effects in the prefrontal cortex were stable. But then what about adults? So we can ask if there's still hope for us if we're looking to kind of change our brains. So we can try to answer this question using data from the Resource Project, which collects data over 3 different types of mental training. So there's first this presence type of training, which focuses on attention and interoceptive awareness through breathing and meditation and body scan type of exercises, sort of like mindfulness. Second, we have this socioaffective training that aims to enhance dealing with difficult emotions and compassion for the self and others. And there's also this more sociocognitive module that focuses on perspective taking regarding both the self and others, again, through the concept that people can take on different roles and being more aware of these different roles that people can take on. So the training operates over the course of 9 months. So the first 3 months, both cohorts of about 80 people completed the presence training, And this is followed by a crossover design of socio-affective and social cognitive training in both groups, also across 3 months. There's also a third cohort that only completed the socio-affective training in the first 3 months, and this is this active control group. And then there's also these retest controls that didn't undergo any training but that were tested or scanned at multiple intervals. So this leaves the study with a total cohort about 330 subjects. So this is work that was led by Sophie, and what she wanted to understand in this work is to understand how brain structure and function change as a function of these different types of training. So she used these a priori networks that were extracted from neurosynth, and she specifically hypothesized that network markers in these, or microstructural markers in these different networks would change and follow the different types of social mental training that were completed by the participants. So this would mean that attention and interoception type of networks would be more affected by presence and attention training. Emotion and empathy networks would respond to social affective training, and these theory of mind networks would respond to the perspective taking module. So to study how brain microstructure changes as a function of these 3 types of training, the study leveraged quantitative T1 imaging. So as I presented earlier, this study used these kind of depth varying microstructural models to measure microstructure and as a proxy of laminar elaboration in these different networks. And we can see that the different networks that were mapped here show these diverse profiles of these diverse microstructural profiles, but we just generally see these higher myelination values in deeper layers, although there's some variation across the different networks. And normalizing these intensity values per depth really emphasizes these cross-network differences further. So how did the different training schemes reshape network microstructure? So what we want to focus here, if you can see, is how these colored lines diverge from the 0 line, which is marked in black. And one thing that is quite striking here is the diversity of the patterns of change across these networks and training modules. So for instance, changes in perspective taking, so this green line, related to increase in myelination in superficial compartments in almost all networks, with less change in deeper compartments. But compared to other training modules, the perspective training module was associated with the strongest increase in myelination in the network that was associated with theory of mind. Also, affect training, so these red lines here, seem to have a diverging effect from the other modules in the emotion network, where all depth compartments were shifted, with again strongest effects seen in superficial layers. And what was interesting also is that these changes associated with presence training reflected an increase in quantitative T1 values, suggesting a decrease in myelination in more superficial compartments. So this could be due to retest effects, although the corresponding effects in just the retest controls were weaker than what was observed in response to the presence training, so interpretation here is not so straightforward. So there's some evidence, at least from these results, in favor of some domain-specific changes in microstructure associating these networks to their corresponding training schemes. And on a domain general level, so changes over the 9-month testing period, Sophie found that myelination in these transmodal cortices was particularly susceptible to change. This was particularly seen in these middle and deeper compartments, and these patterns were observed in both training cohorts, but not in controls. So to summarize, there seems to be this mix of more domain-specific and domain general effects on these mental training modules on cortical myelination. So some patterns of quantitative T1 change seem to be domain-specific and vary as a function of cortical depth, and overall changes were largely observed in transmodal deep cortical compartments in both cohorts over the 9-month period. So just to conclude, we think that myelin can help us understand the effects of environmental demands on the brain. So I presented some findings from Sophie's group that showed that maturational trajectories of cortical microstructure are associated with adaptation to changing environmental demands in youth. And even in adulthood, there seems to be some domain-specific changes in microstructure that follow different types of mental training. So overall, these results offer us some promising insights to try and integrate social factors and neurobiology. And although there's still some challenges that remain to be addressed here, notably regarding the necessity for really high-quality longitudinal data, as well as well-phenotyped samples to make these associations between social environments and changing myelin. So with that, thanks to all the people that actually conducted this work. And yeah, thanks for being here.